Hi, so this is the lecture for Chapter 3, Active Cellular Transport. So this is the last part of it. So I am actually going to review a little bit. Um, I'm going to review a little bit with the cell membrane. So the cell membrane is a phospholipid bilayer, right? So this part is fat, so it's hydrophobic. It does not like water. That's why it folds to the inside. And the phosphate heads have a charge to them, so they like water. So they're polar. Polar likes polar, and water is polar. And our cells are actually bathed in water. And so that's the bilayer. And so all cell membranes are made up of this bilayer: the nuclear membrane, the endoplasmic reticulum membrane, the Golgi membrane, the vesicles. All the membranes in your cells are actually made up of this phospholipid bilayer. So this is an important membrane. And so, and it's fluid, which means it can move around. So it's very fluid. And the cholesterol here actually keeps it fluid. So if it gets too cold, um, the cholesterol actually keeps it sort of stable. If it gets too hot, the cholesterol also keeps it stable. So cholesterol is really important for the stabilization of the cellular membrane. But it is called mosaic because it has all these different types of proteins and lipids and glycolipids and glycoproteins attached to it, right? And so also the cytoskeleton and stuff is attached to it. So it gives it structure, helps it stay in place. And so this is a glyco. Glyco is sugar, right? And then this is a carbohydrate. So this is all of these green things sticking up here are carbohydrates. So you have a glycoprotein. That's a carbohydrate, and then you have it that's attached to a protein there. And then you also have it attached, instead of the phosphate, it's attached to that phosphate head. So that's a glycolipid. And then you have proteins, and they do different things. So I would know those different proteins. They're general, right? And so if we were in class, we would have made a model, and this is one model, right? So here they made their phospholipid bilayer. Here's the phosphate heads. Here's the cholesterol. Here's a proton pump, which would actually require energy. Here's a transport protein. This could be like an aquaporin. And here's a receptor protein, which actually could be similar to what we're going to talk about with the COVID-19. So that ACE2 receptor protein actually, but there's could be different signals and things like that. So what else is on here? Oh, here's a glycolipid, right? So the carbohydrate is attached to the phosphate head. Here's a glycoprotein where it fell off, looks like, but the carbohydrate is attached to the protein. And here's a peripheral protein, anyways. So here's another one that I had that they did. So that one was similar. Okay. I am going to review this a little bit. And during the classroom, I did talk about the diffusion lab. So the things you have to keep track here is the cell and the solution outside the cell. And in the lab, it gives you the bags, the semi-permeable membranes. And so that takes the place of the cell, and then you have the solution outside of it. So hypotonic, so water is the most hypotonic you can get. So water is pure water. If there's nothing in it, is as hypotonic as you can get. So that's the highest concentration, and then we're talking about the solution here. So the solution is outside the cell. So if you have and it's going to move down the concentration gradient from high to low. So it's going to move from high concentration to low concentration. And water is going to move into the cell. That means the cell is going to gain water, gain weight. And in animal cells, it's actually going to lyse our cells or break our cells open. They'll burst, right? So if you're an animal cell, you don't like this condition. If you're a plant cell, though, you have a plant cell wall. And actually, the plant cell wall, that the water inside this vacuole gives it pressure, and it pushes up against that plant cell wall, and it keeps the plant upright, right? So if you don't water a plant for a while, it's going to lose water in this vacuole, and but eventually the plant cell will wilt and die. So this is animal cell. This is a plant cell. Okay. So plant cells like it in a hypotonic solution. They like it in pure water. Our cells do not. Our cells actually prefer isotonic. So that when you have an equal amount of water inside and outside the cell. So it's the same. And so water can move in and out of the cell through that aquaporin protein, but it cannot, there's no net gain, right? So there's an equal amount of water moving in and an equal amount of water moving out. That's what we like. And plants can survive here, but they prefer this. So this is a hypertonic solution. So the solution out here has less water here and more water inside. So this could be sugar water or salt water, or but it has to be pretty high amount, because in that case water is going to move out because there's more water inside the cell than outside the cell. So water is going to move out. And so that means that's going to shrivel. In this case, the plant is going to die as well. So the cell membrane is going to pull away from that cell wall and it's going to die. So this is the condition that animal cells like. This is the condition that plant cells like. So, and again, but here the cell is going to gain weight. Here the cell is not going to have a weight change. And here the cell is going to lose weight. So that's what you have to keep track of. So you have to keep track of the solution and the cell. And in the case of this, it's the semi-permeable membrane, the bag. I think they call it dialysis tubing here. So you have to keep track of the solution in the beaker, and you have to keep track of this and which way the water is going to move, right? So this is a semi-permeable, so it lets small things through, but it does not let the bigger molecules through. And sugar is a bigger molecule, which is what they use in that lab. So sugar cannot move through the membrane. Okay, so hopefully that helps with that. And so this is the aquaporin, and water cannot move through the membrane this way because water is polar and this is nonpolar and it won't let it through. So it actually uses this protein here called an aquaporin, and this is how water moves across the membrane. So it's passive transport, right? So it moves, which if there's more water outside than inside, water is going to move from high concentration to low concentration. If there's more water inside the cell than outside the cell, it's going to move that way. So it can move both ways. And the way this protein is folded is the nonpolar R groups will be out here, and the polar R groups will be in the inside. So it's important the way this protein folds, basically. And this is what we did last year, and this is what we would have done this year if had been in class. But last year I had 4 periods, and one did a carrot, a potato, an apple, and a sweet potato. And what they did was they took, cores of each vegetable or fruit, and they weighed it, and then they put it in a cup with solution in. And these are the 6 different treatments. So they put the cores in pure water in 0.2% sucrose, 0.4%, 0.6, 0.8, and 0.1. So this is the most hypotonic. This is the most hypertonic solution. This is solutions right here, right? And then the potato was the cells. Potatoes have cells in them. So anyways, so then they let them sit for 24 hours, and then they came back, and then they weighed them again. And so if they gained weight, right, so this is the 0 line, this is the isotonic line, this is 0. So if they gained weight, they would be above 0. And if they lost weight, they would be below this 0, right? So this is water. This is the most hypotonic solution. Water will always move in, and the sweet potato should gain weight. And that was true for all of these, right? At 0, most hypotonic, they all gained weight. And then this is the isotonic point. So this would be about 0.5 for a carrot. So that is where there's equal amount of water inside and outside the cell. So this is the isotonic point, and I would have had you do some water potential problems with this, but the isotonic point here for the potato is about 0.4. The isotonic point for the apple was 1. So these are sweeter generally. And the isotonic point for the sweet potato was about 0.7 sucrose. So anyways, I just wanted to show you that. But if it's above the line, it's hypotonic. Right here is the isotonic point. And then if it's below the line, it lost weight, which means it was in a hypertonic solution. Water moved out. So I just want to sort of explain that a little bit. That goes with that lab. Okay, so I'm going to, for this one, this is active cellular transport. So we're going to define active cellular transport, describe how primary and secondary active transport moves molecules across the cell membrane, describe how vesicles are used to transport substances into and out of the cell, and distinguish between the forms of endo and exocytosis. That's vocabulary. Okay, so we'll review passive transport. Passive transport moves from high concentration to low concentration, and it does not require energy. So it requires a gradient, right? So if there's equal amounts of sugar on each side, that would be isotonic, and water can move back and forth, but there's no net movement. So this requires a gradient. It has to have a high and a low concentration, but it does not require energy. Okay, so however, not all molecules that are needed by the cell exist in high concentrations outside the cell. In these cases, the particles need to move from an area of low concentration to an area of high concentration or against the concentration gradient. So the way you can think about passive transport is it's flowing downstream, right? So if you get in the water and you just sit there, you're going to flow downstream. So that's passive transport. Active transport would be swimming upstream. So it takes energy, and that energy will be in the form of ATP. So so here you can see there's no solute here, and there's lots of solute here. And a passive transport, it would move from high concentration to low concentration. But this is active transport, so they're moving it against the concentration gradient. And if you swim upstream, it's gonna require energy, and that energy here is in the form of ATP. So I think that's what I just said. I think I just said all that. Pretty sure I said all that. And this is the ATP molecule. And if you just look at this right here, this is actually a nucleotide. This is the adenine nucleotide. So it has the phosphate head, the ribose sugar, and then this is the nitrogenous base. So they actually think that DNA evolved from ATP. That's what they think. So this is actually a nucleotide right here. So if you add 2 more phosphates to this adenine nucleotide, you get ATP. And if you notice, these phosphate groups have charges on them, right? Which makes them high energy. These negatives do not like to be together. And so if you break this bond here, it will release energy. So this is ATP, and it's at adenosine, that's this part, triphosphate, TP. So that's TP, right? I think it's on the next one here. I'll show you. Okay, yeah. So this is adenosine, which is this right here, this part of the molecule, and then triphosphate. Tri is 3, right? So that's 1, 2, 3 phosphates. When you break that bond, it's going to produce free energy. So this is ADP, diphosphate, right? So it lost a phosphate, and it has an inorganic phosphate. So it's inorganic because it doesn't have carbon or hydrogen attached to it. So you're going to take this is potential energy, and this is energy that is used, right? So and this is actually renewable energy, and ATP can be made somewhat in through photosynthesis, but you make your ATP in the mitochondria. And so basically what happens in the mitochondria is you take this ADP molecule and you're going to use your energy from food, which is basically glucose, gets fed into the mitochondria, and you're going to attach this inorganic phosphate to ADP to make ATP. So this is potential energy, and this is the energy currency of your cell. This is what your cells identify as usable energy. And then when they use that energy, they're going to break this bond, this covalent bond right here. That energy is going to come out, and that's going to provide energy for cells. And now you're back to ADP, and then it's going to go back to the mitochondria or photosynthesis or the chloroplast, and that's going to get attached again, right? And you can actually take two of these off. I think that's called AMP. But I would just understand, that this is ADP and I would know the difference between these two. This is ATP, T is tri, potential energy. This is energy that is used. Okay, there are three types of active cellular transport: molecular pumps, endocytosis, and exocytosis. So I'll go over those. Okay, so outline two differences between passive and active transport. Name the three types of active transport. Name the energy-rich molecule active transport primarily utilizes as an energy source. That's ATP. I just went over that. But passive transport goes along the concentration gradient. So this is swimming downstream. And in active trance, they travel against it. So this is swimming upstream. Passive requires no energy, and active does. Passive transport also requires a concentration gradient. So if there's no concentration gradient, it doesn't move. The solute won't move or the water won't move. Three types of active transport: molecular pumps, endocytosis, and exocytosis, and ATP is the energy-rich molecule. And again, if you show this is moving against the concentration gradient. You'll know it's going to be active transport if it's moving against the concentration gradient. So there's more down here than up here. And also if you see this ATP, those are two giveaways that it's active transport. So that's what this is showing you. So it's moving against the concentration gradient. So this is low to high. It's moving against the concentration gradient, and it requires ATP. So ATP is bonded to the protein. So generally ATP has to be bonded to the protein before it works. The ATP kind of turns it on. This phosphate will actually turn it on. There's better ones than that, but anyways. Okay, so this is the sodium-potassium pump. I'm going to actually show this one here. It does require ATP, and so it's moving sodium against the concentration gradient, right? So there's less sodium here and more sodium up here. So it's moving against the concentration gradient, and it's moving potassium against the concentration gradient as well. So there's more potassium here than here. So it's moving it up the concentration gradient. It's moving against the concentration gradient. And I'm going to show you this one here. So here, it's going to move this. This is a transport protein in the lipid bilayer. And it's going to open this way. Those sodiums are going to attach. And then this ATP is going to attach, which is going to turn it on, right? And when it gets turned on, it's going to open the other end. It's going to release those sodiums out into the extracellular. So this is inside, right? This is inside the cytoplasm. This is outside the cell. So it's moving sodium outside the cell. And then it's going to come in and it's going to have potassium attach here. And then that phosphate group is going to move off, and then it's going to have to go back to the mitochondria to get attached back to make ATP. And then it's going to open back up into its original form, and it's going to release that. So it's actually moving, and this is called phosphorylation. So phosphorylation is when you gain a phosphate and you get that phosphate from ATP, which then makes ADP, right? And this is considered an inorganic phosphate. That's what that is. Okay, so that's that. So this is cotransport. So it uses the cell's electrochemical gradient to move molecules into the cell. As ions move into and out of the cell, they create differences in electrical charge across the cell membrane, forming the electrochemical gradient. This process is known as cotransport. So this is generally done with protons, and protons basically are hydrogens that have lost their electron. So these are protons and these are charged. But again, this is a proton pump, and I'm not sure if these work or not, but this is low concentration to high concentration. So it's moving against the concentration gradient, so it's going to require ATP. So it's swimming upstream, right? So it's going to require ATP. And these are charged, which gives them that, so now this is more negative and this is more positive on this time. And you have a gradient here. And you're actually going to use this gradient. So this is going to supply the gradient, and this requires ATP. But now this is going to move with the concentration gradient, right? So this is 8. I showed you back up here, right? So this is more plus, this is more minus, and it should flow through diffusion. It should want to flow the other way. So now it's going to use this different protein to actually flow back this way, but it's actually going to take this big molecule with it. And so that's why it's a co-transporter. So this creates the gradient, which requires ATP, and then it uses this gradient to actually transport this sugar back inside the cell. So it's called a cotransport. Okay, so I think I already did that. I'll skip that. Okay, so these, so this is the electrochemical gradient, and again, this is done with protons. So to create the gradient, it requires ATP, it pulls it across, and then this is the cotransporter. This is what I just showed you. All right, so you have symports and antiports. So an antiport will make them go opposite ways, right? So this is pumping sodium this way, and this is pumping potassium this way. So this is the sodium-potassium pump. This requires ATP, right, to turn that on. So when it gets phosphorylated, it turns it on. When you take that phosphate off, it dephosphorylates it. And then this is the symport where they're both moving both ways. And so it's moving sodium and glucose both ways. But there has to be a gradient here. It has to create a gradient in order to carry that glucose with it. Okay. So we'll go to this, okay. So primary active transport directly uses ATP to bind to the pump, while the secondary uses the electrochemical gradient, which is generally done with hydrogen, but not always, with those ions, or other alternative energy sources. So you can make gradients with different things, but a proton pump uses those H pluses. Okay, so I'm not going to go over that, but I think this is the better one to use here. This is, I think, a better one to use. This one here. I would look at this one here. This one explains that, right? So when this here gets phosphorylated, this protein changes conformation, right? It opens on the other side. And then when you take that phosphate off, it goes back to the original thing right here. Okay, we'll do vesicle transport. And again, so these are vesicles, and this is important in the endomembrane system. There's all kinds of different vesicles. This is a phospholipid bilayer, right? It has a bilayer. Then it has something inside of it, and then it has the surface. But like as things move from the rough ER, the smooth ER to the Golgi, that moves in vesicles. As it moves from the Golgi to wherever it goes, that generally moves in vesicles. So there's lots of different types of vesicles. But some molecules are too large to move directly through the membrane or make use of membrane-bound transport molecules. The cell has two other methods of active transport, which requires ATP. So endocytosis moves large molecules into the cell. So this is endocytosis. So it's moving in, right? So it's taking stuff from the outside and moving it in. And exocytosis moves large things out. So this is exocytosis. So there's three types of endocytosis. There's phagocytosis. This is food. Pinocytosis is liquid, and receptor-mediated cytosis. This is actually how it brings in the coronavirus. Okay, phagocytosis is a cell eating. So these leukocytes engulfing and then destroying pathogens and protists engulfing solid food particles. So they come in here and the cell membrane starts going around it, and eventually it makes its own vesicle right here. So that's how it moves in. And then if this is food, then it would hook up with the lysosome. The lysosome would break all that stuff down, like into amino acids or simple sugars or lipids or something that the body needs. Nucleic acids, it can break it down into nucleic acids for brought in cells. This is cell drinking, and again, it forms a vesicle, but these are mainly liquids. And they create much smaller size vesicles. But anyways, this is cell drinking. And then the receptor mediated, the example I gave you on here was the coronavirus, right? So the coronavirus has that spike protein, and it's actually going to land on that ACE2, that ACE2 receptor protein. And that's kind of like a lock and a key. And then eventually it's going to get taken in, and the video shows that pretty well. It's going to take you in there. So if I ask you on a test how the coronavirus enters your cell, you would say receptor-mediated endocytosis. That's what you would say. And that's what this is. That's an overview of that, right? So this is endocytosis. This is food particles, this is liquid, and this is receptor mediated. Those are the three types of endocytosis. And this is my graph I put on there, right? So this is phagocytosis, and this is pinocytosis. So this is food particles, and this is liquids. And it shows you there. So endo is in, right? And exo is out. So exocytosis is how you get rid of big things, right? And this is going to come up and it's going to fuse with this cell membrane. So this cell membrane is going to fuse with this cell membrane, and then it's going to release it. All right. So again, this is all fluid. These are proteins here. And so so this is coming from the Golgi actually. And so if this contains a protein that's needed outside the cell, and right here it has a label on it, right? So some kind of label. So this is a protein here. It's probably going to go on the cell membrane right here. So, or plasma membrane, they're the same thing. And then here's another one coming from the Golgi, and this has a signal on it that's going to go outside the cell, right? So it's calcium triggered. So there's all kinds of, so the Golgi gives it signals and say, okay, this one needs to go here, this protein needs to go here, this lipid needs to go here. And they actually sort of tag them, and so they know where to go. It's actually like a little factory. Okay. Okay, name the organelle which is used in the endo and exocytosis. Name the 3 types of endocytosis. Describe briefly how the 3 types of endocytosis move molecules, and describe how the cell uses exocytosis. So basically, they use those vesicles, and they have those phospholipid bilayer membranes. The 3 types of endocytosis are cell eating, cell drinking, and receptor-mediated cytosis. This is how the coronaviruses comes in. So that's what that says. And then exocytosis, it gets rid of waste products, it gets rid of proteins that are needed outside the cell, things like that. And I think that's it. So that's it for this one. And you need to fill out the guided notes that go along with this, and I hope that's helpful.